I'm Dave Kramer, the Program Director for Planet Texas 2050. Really excited to have you all here, and I'm glad there's such a group. You never know how many people are going to make it at lunchtime on Zoom when we're all zoomed out. But as part of our fall showcase, we're trying to just present and have the researchers who are involved in the Planet Texas 2050 network present the amazing work they've done and are continuing to do. You know, Planet Texas 2050, our main aim is to make Texas more resilient in the next 30 years, knowing that we have only a certain amount of time to get ready for all the changes that are happening around us, and particularly related to climate, population change, and all these other related drivers. But today's event, we're going to be hearing from a couple of different teams. The first is, I believe you all are going first, right? Katy Brown and Michael Shensky on a project called Geospatial Modeling for Applications Addressing Infectious Diseases. And I never quite figured out how to pronounce Burkholderia, if that's the proper pronunciation, but it's a really interesting project looking at how infectious diseases are moving as climate changes. And you can do way more justice to it than I can, but in a nutshell, we're looking at, the team has been looking at how we can map and prepare for dealing with these changes around us. And then the other project that will be presented today by Anna Dabrowski is the data stewardship and DOLCe Initiative, the Digital Object LifeCycle Initiative. Basically, an initiative to try to ensure that we have the necessary infrastructure, mechanisms, and policies to allow data to be findable, accessible, interoperable, reusable, or fair at the end of a research life cycle, and something that can help all of the different projects, both in Planet Texas and beyond. So without further ado, I'll stop there and let each team do much more greater justice to their work. So thank you so much.
Okay, I think I'm starting. Let me see if I can share my screen. Does that look okay?
Looks great, yeah.
Okay, so my name is Katy Brown, and I'm going to be splitting the presentation with Michael Shensky from the libraries. And we are going to talk about a project which really was initiated on a specific organism, but actually has broader applications to general issues related to the spread of infectious diseases. I will have a kind of Texas-centric section, but it's important to understand that this is not a Texas-centric problem, it's a global problem. And I want to begin by actually saying that this is really a team effort. And I've been so privileged to work with such a really fantastic set of team members. Michael Shensky and Jessica Trologan from the libraries who've really been helping drive the GIS work, which Michael will show in the second half of this presentation. Stan Rue, who I work with quite closely in MBS, and he brings in elements of crop science and plant molecular biology, which we won't be discussing here, but are actually quite important in understanding the spread of these diseases as well, particularly because the one I'll be focusing on today is a soil-based disease, and it is quite related to agricultural as well as urbanization. A few other people I want to mention along at UT, Donna Bruhis, who I know is on here. She's joined us recently as a capstone student project, and she is a senior in environmental engineering, so she's beginning to work with that. Both Stan and Tim Beach, who are helping with the supervision of her. Tim brings in some interest, also interesting background in soil work, which is really good. We haven't quite connected with him yet, but he is listed as a supervisor, so we will be engaging with him soon. and Tim Keat, who's also on the line too. I know he's tuning in today, and we've already been interacting with him about overlaps between our project and his interest in looking at vector-borne diseases as well. Again, though, this work has really relied on the enormous support we've had from TAC, with Suzanne Pierce and her team. And Anna also, who's on the line, has been directly involved in the project along with Jamie and Lissa. And really, to make this project work, again, this is part of also beginning to move our ideas into the DataX portal, and also with a broader view of making TAC a portal for displaying and accessing this data, not just to Texas, but on a global level. So, so that's kind of the grand view. And finally, I need to mention Ben Butler, who's my postdoc based in Cambridge, who's also started to work on the project as well, and beginning to work on, we have a related project which we're in the process of hearing whether we'll get funded on, where we take some of these tools that we've been developing Planet Texas and working with the government in the Malawi, again, looking at infectious diseases and geospatial mapping to help there. And that's something for another talk. So, so I'm going to set the broad picture and explain how we ended up looking at this in Texas. So there's certainly been recognition for quite a while that climate change is impacting on health. And one of the best places to go to see this discussion is the WHO website, which describes several reports and policies that are attempted to be implemented with regard to this. Now, if you see that according to WHO, what's very important is that all populations will be affected by climate change, but certainly some populations are going to be more vulnerable to it than others. And they cite children, elderly people, people with infirmities or pre-existing medical conditions in areas with weak healthcare infrastructure. Now, when this was written, this was pre-COVID. And I think what COVID has done is shown that these vulnerabilities are not necessarily associated with poor and developing countries. All countries have been exposed to have these vulnerabilities. And in a sense, it really emphasizes that the effects of climate change are not just limited to impoverished areas or vulnerable communities in what we traditionally think is impoverished areas, but both the developed and the developing world. Now, according to them, they anticipate 250,000 additional deaths will come by 2030 based on climate change. And they quote some direct costs as well, so billions of dollars per year associated with this. So one of the drivers and why we're interested in looking at this. So we do know that climatic conditions strongly affect, in the area of infectious diseases, water and vector-borne diseases. And I should add that I've been working in the area of neglected tropical diseases since the mid-90s. So, and a lot of my work originally was really based in the tropics, where I've spent a lot of time out there trying to come up with diagnostics and therapeutic in resource-limited settings. But really what's been interesting is as time has progressed, we've seen these tropical diseases moving north and south of the areas traditionally considered the tropics. And climate change, certainly according to WHO, but many other sources, are likely to lengthen transmission seasons, alter the geographic range of different sorts of vectors, and as well as microbial-borne diseases like the one I'm looking at. Examples cited by WHO are malaria, where the mosquitoes are already known to be strongly influenced by climate, not a disease that's yet impacting in Texas in any significant way. But another mosquito, the Aedes aegypti mosquito, which is the vector of dengue. It's also highly sensitive to climate conditions, and it suggests that climate change is likely to increase exposure to dengue. Now, dengue is one of the diseases which we are beginning to see in Texas. That's been in the last 5 years or so. And what's really important to know about dengue, it's at the moment, compared to malaria, it's the most rapidly expanding vector-borne disease in the world, and also very one of these which is also under-recognized as a neglected tropical disease. On the right was is a map that Michael Shensky produced that just gives you a sense. This was produced about a year ago, showing that as a result, this is just changes, average changes in temperature over in January 2019 compared to, I think it must be a 30-year base prior to that. And you'll see that a lot of times people are thinking about areas getting hotter, and some areas are also getting colder. And again, this is affecting the spread of diseases, both the vectors as well as the as diseases which can expand in the environment, including soil and water. So what about Texas? So Peter Hotez has been in the news a lot about, he's been a big spokesman both on national and international news about COVID. He himself has spent a lot of time working on neglected tropical diseases, and particularly viral diseases. A couple of years ago, he wrote a nice paper titled "The Rise of Neglected Tropical Diseases in the New Texas." And this is something, the concept of New Texas, something certainly that Planet Texas is addressing, where we are now and where we are in the future as well. And in this very nice paper, he talked about major external factors driving both emerging and neglected diseases in Texas: population expansion, human migration, urbanization, poverty, climate change, absolutely, as well as declining vaccinations and transborder traffic and sea transportation, which are all listed here. Now, with regard to climate, he points out that Texas is disproportionately at risk to global warming and cites a few studies. And that the projection of days over 95 degrees Fahrenheit by the 2050s is expected to be on at least 100 days compared to 40 such days now. And these warming effects are particularly conducive to the expansion of NTDs. And again, on the right-hand side is a picture that I've taken out of his publication, which shows where the most areas of geographic concern within Texas lie. Now, this has been particularly focused on vector-borne diseases, so diseases transmitted through mosquitoes and sandflies. I myself worked on leishmaniasis for quite a while too, and West Nile virus, which I'm sure most of you are aware. We've had West Nile virus again in Central Texas. And down towards South Texas, the border, where we're seeing, as I mentioned before, dengue, but other mosquito-borne diseases as well as other vector-borne diseases. So this is not a thing of the future. This is now. And the expansion of these diseases into Texas is certainly something that's a more recent event rather than something that we've had for many years. Let me see. Oops. I don't know why that's not responding. Oh, there we go. So I'm going to go now talk about a little bit about melioidosis, which is the disease that we initiated our studies on. And we initiated this primarily because it's a disease I've worked on since the mid-90s. And when I'm partly based in Austin, and a lot of my fundamental research here involved developing diagnostics and therapeutics for melioidosis in Texas, built on previous work I'd done in the UK on melioidosis vaccines, which I can say overall still have not been very successful. It's a very difficult disease to develop vaccines against. But just to give you a little bit of a background, so as I said, I started working on this disease in the mid-90s, and it's caused by a soil and water-borne bacteria known as Burkholderia pseudomallei. It's a highly neglected tropical disease and highly fatal throughout the tropics. Now, originally the disease was thought to be primarily endemic in Southeast Asia and Northern Australia. But as you'll see later, once people began to realize that environmental conditions are suitable throughout the tropics, it's one of these diseases that once they started to look for it, they began to find it. And it's mainly acquired through skin, or through inhalation. And the typical way is associated with agricultural areas where you have a lot of contact with water and soil. So the traditional way of thinking about it was around rice paddies, and certain northeast Thailand has been an area, a big hot spot for many years. But there are a lot of hot spots. It just happens to be that it was well recognized in Southeast Asia. One of the big problems about melioidosis is it looks like any other flu. You can have pneumonia, but you can also have abscesses on the skin, though not necessarily in the pneumonic form. You get septicemia, and mortality rates are very high, 40%. If you think about the mortality rates of COVID, although we know COVID is a very deadly disease and it's very widespread, mortality rates of 40% are extremely high. And if you get an acute infection, you can be dead within several days. Part of this is the fact that although it can be treated, the organism is highly drug resistant. And so one of the problems is if you don't recognize that this is the disease, you'll treat it with the wrong medication. And also there aren't very many, there's no licensed diagnostics, there are no vaccines. And when you do get diagnostics, it's often slow and inaccurate. In fact, when I used to talk about this, people were rather surprised. But I think now in our current stage with the pandemic, these sorts of situations are not so unusual, and we can see the effect of them in right now in terms of no vaccines and slow and poor diagnosis. However, I do want to say historically, Michael and I wrote a little piece for Earth Day for PT 2050, and this disease was not unknown to physicians in US physicians, particularly during the time of the Vietnam War. Time Magazine had an article about it, referred to it as Vietnam's time bomb. And you'll see I pulled out a little quote there, but you'll see that this was just early on during the Vietnam War and when the disease was just being recognized. But you'll see that here they talk about 32 servicemen and 9 dying, which is a rather high percentage. But one of the big problems is it also has the ability, a lot like tuberculosis, to remain dormant for many years. So you can contract it, and many years later, when you become immunocompromised through age, diabetes is also a factor, the disease reemerges. And as I mentioned earlier, it can occur within a few days to weeks. So, melioidosis in Texas. So it's interesting. We proposed this project back in, I believe, early 2019. And that was partly because I had a recognition that that the disease was already spreading. And it was earlier that year or the previous year that there were some reports of the disease in northern Mexico in latitudes that were similar to South Texas, South Central Texas. And so we were very fortunate to get a little bit of seed funding from Planet Texas to start this project off. And it enabled us to start to begin to explore some improved map predictions. And this was one of our predictions that Michael Shensky, our early predictions, Michael Shensky helped generate with us, that we presented back in October at the World Melioidosis Congress. I should say it was the only map there other than one map that was produced about 4 years before that was in the literature. And much to our surprise, maybe not surprising, but it was a bit of a surprise to the audience. Later during that conference, the CDC got up and announced that Texas was now the first mainland state in the US to be endemic for melioidosis. That was in October last year. And June just this year, they finally published the case, which was where they truly believe the disease was acquired. As I said, this is only one case. And one of the reasons I say that the disease is really probably poorly documented is simply because it's not been recognized. So that means that there are probably people acquiring the disease. We're just not diagnosing it appropriately. So this brings me to the starting point before I switch over to Michael, which was the starting point really emanates from 2 pieces of work that were published around 2016. One is this predicted map. And this was the only published predicted map about where melioidosis could be in the world. It was done by a really leading scientist based out in Thailand, collaborating with some geospatial experts. And what you'll see is we have really just the one map. And although down there, you can see a little bit of Texas and Florida appear in the prediction. But an important point about this, which is often a big issue with these geospatial maps that we see for infectious diseases, is that almost the only information that you can get is a published map like this, and that's all you have to work on. They themselves are very difficult to work with, and in this case, there are very, very many parameters that were put in to generate this map, most based on what is known about the disease in the most endemic areas of the world. So based on parameters that describe the environmental conditions primarily in Thailand and Northern Australia. But as you can see, looking at where the disease is known and also is could potentially be, not everywhere in the world looks like Thailand or Northern Australia. And there was a second paper that appeared in Nature, just a comment about the global picture of melioidosis by good colleagues of mine, Bart Curry and Miriam Kestley. And Miriam is probably the leading ecologist in this particular organism. And the pictures shown above are on the left is a typical rice paddy in Thailand, and the typical area where you might isolate Burkholderia pseudomallei in Northern Australia. And the point they're making is these ecological niches look very different. So the picture I show below again comes from the Hotez paper, and this is of a colonia in Southeast Texas. Again, showing a lot of the risk factors that are associated with acquiring this disease and many neglected tropical diseases, which are poverty, poor housing. You'll see the set of tires, which are well known for collecting water and permitting the expansion of mosquitoes. Wet soil, again, and floods, which are often associated with the spread of this disease. We see a cyclical spread in monsoon season. And it brings my final point here is that when you see one of these diseases, such as melioidosis, it's often associated with many other neglected tropical diseases that survive in the same environmental conditions. So it wouldn't be surprising if we started to see another outbreak of dengue in South Texas, that those environmental conditions are also permissive for diseases like melioidosis and other vector-borne diseases to expand. So I'm going to turn it over to Michael to show how we're trying to peel apart part of this data from this, based on this singular study, to make the data much more accessible, interpretable, and to give us more options in order to study the spread of this disease and other infectious diseases based on climatic and environmental factors. So I am going to, maybe Michael, you want to go ahead and share your screen?
Yep. Thanks, Katie. All right. Can everyone see my screen?
Looks good.
All right. Thanks. All right. So thanks, everybody. And thank you, Katie, for that introduction. And thanks, everyone, for joining us today to learn a little bit more about our project. I don't have much time, so I'm going to go very quickly, but hopefully this gives you an idea of what we've been working on in the last year. So I just quickly wanted to show basically the data set that is really at the core of everything we're doing. And this is the data set that lists documented occurrences of Burkholderia pseudomallei in soil or water, or melioidosis infections in humans or in animals. And we can see examples of what these actual records look like. So we have the type of occurrence, what country it occurred in, and we actually have the coordinates for those locations, which are really important for the geospatial work we're doing. So what we did is after we attained this data set, which is freely available online, it was generated by a particular researcher and incorporated into the project that Katie was talking about earlier, where that map predicting suitability for melioidosis was developed back in 2016. We're using that same data set, but what we're trying to do is bring in some additional other data that we can use to try and determine what the unique characteristics of these locations are on Earth where Burkholderia pseudomallei has been found in soil or water. And so what we decided to do is bring in soil data, climate data, that contain information about variables that prior research on Burkholderia pseudomallei has found to be correlated with presence of the organism in soil or water samples. And so we are using data from soil grids. We're using the Harmonized World Soils database. We're using some of the data from the WorldClim data set that is available. And these are all raster data sets. And so using these raster data sets, which we see an example of there at the right, that's one of the soil grids data sets that we are using. We can see that if we zoom in, we have these pixels that contain information about the values of a particular variable at locations all over the world. And so what we're trying to do is look at certain variables, again, that have been found to be positively correlated with presence of the bacteria in the environment. And we're trying to automate a method for detecting where these locations are and looking at the underlying soil and climate characteristics of those locations, and then trying to extrapolate and determine if there are other areas on Earth that have those same conditions that haven't previously been studied, where the Burkholderia pseudomallei could perhaps be in the environment already, or that environment is suitable for the bacteria. If it was ever introduced there, it might be able to sustain itself. And so in order to carry out this analysis, we're using an automated approach, specifically using Python scripts to automate QGIS processes. So QGIS is a free and open source GIS application. It's used for geospatial analysis and mapping. And so the part I've been working on for this project is writing that code to automate these geospatial processes. The first stage of which, and we can kind of see that code a screenshot that I've taken of the actual code that does most of the work here is in the background. The first part is really identifying which of these cases from that larger spreadsheet of about 22,000 records actually document presence of the bacteria in soil and water samples, where we know exactly where that bacteria has been found in the local environment. Again, making some assumptions here about the accuracy of those coordinates that are provided in that spreadsheet, but we should know that these are the environments that it was found in. The other yellow cases that we see on this map are cases where melioidosis is being diagnosed. There it's a little bit trickier because, as Katy has mentioned, you can pick up the disease in one place but not show symptoms until you're perhaps on the other side of the world. And so it's a little bit difficult to assume that those cases were actually acquired in that specific local environment. Whereas the other points marked with the purple dots, those we can assume that this is where the bacteria was found in soil or water. So those conditions in that immediate area are reflective of where that bacteria can survive. So here, this is a graphic representation of basically what the code is doing. And we're taking these separate data sets, again, representing soil and climate variables. So things like annual mean precipitation, the percent of clay in the soil as opposed to silt or sand, and soil pH, looking at the acidity of the soil. We're looking at where these occurrence points, which are marked with the white triangles, what the conditions for each of these variables are at each of these specific locations. And again, looking just at variables that have been found to be correlated with presence of the organism in soil or water. So once we've determined the range of values that we see at these different locations. So for instance, if we look at all 320 something locations where we've seen Burkholderia pseudomallei in soil or water, and we know that the range of pH values that has been shown to tolerate are maybe 4.5 to 7.2, we know that, all right, we've seen this bacteria survive in this type of soil environment somewhere in the world. So we can assume that it can survive in similar soil conditions elsewhere in the world if those same conditions are present. And we're doing that not just for soil pH, but the other variables as well. So then the next step is really to combine all that information. So we want to see what are all of the areas in the world where we don't just see the soil pH conditions being met, but the soil pH conditions, the rainfall conditions, the percent clay in the soil conditions, where are all of those conditions being met? And so the first product that we generated was kind of a binary suitability raster, where we're looking at all of the locations that meet all of the criteria. So if it fails even one criteria, the soil pH is slightly too high or too low, but everything else is perfect from what we've seen, then that would still be classified as an area where we do not think the bacteria could survive. We then wanted to work towards a more descriptive approach of identifying areas where the bacteria may or may not survive. And so what we did is, instead of just saying yes or no, we think the bacteria can survive here or it can't, we used our coded approach to basically run the QGIS raster calculator to add up each of the number of variables that were found to fall within the range that we established in the previous process. So here we end up with a different result, basically showing how many of those criteria are met in a given location. So in that very bottom layer that we see in this kind of complex screenshot, the red areas would be areas where all of the conditions have been met. Areas that are orange, maybe one condition wasn't met. Areas that are yellow, two conditions weren't met. And so on. And so the green areas are areas where we do not think that the bacteria would be likely to survive. So there are a number of questions we still have to work through regarding this methodology, and things we're still reflecting on, finding ways that we can improve the process by looking at how we handle things like outlier values. How can we deal with somewhat limited sample coverage? Are there ways that we can weight certain environmental variables that seem to be more important based on prior research into the organism and its tolerance for different environmental conditions? So there's still things that we are investigating. It's by no means a totally finished. And one of the great things about this coded approach is it allows us to iterate and improve on the process and make these gradual improvements as time goes on. So I'm going to show you a few examples of our results so far. This is one of the first products we generated, again, predicting where we thought the bacteria could exist. These are the areas that met all of the conditions for, again, soil pH, percent clay, annual precipitation, and a few other variables as well that, again, we selected based on our review of the literature. And we can see that Texas and parts of the southern United States are covered in this predicted area. Here's a slightly more advanced analysis. This is, again, a screenshot that Katy showed earlier, where we see the overlap of suitable soil conditions and suitable climate conditions, giving us the area in purple, where both those sets of conditions are being met. Here's the more complex product I mentioned just a minute ago, where we're not just predicting whether it can or cannot survive, in those stark kind of binary terms, but looking at how many of these criteria are being met, right? So again, these are just predictions, but we see Texas there showing up in shades of orange and red, meaning most of the criteria that we have established for defining whether we think the bacteria can or cannot exist in an environment. Most of those conditions are being met here in Texas. Here's a zoomed in view where we can actually see the county boundaries. And we see, for instance, here in Travis County, most of Travis County, based on this last run of our analysis, is located in an area where all of the conditions are being met for survival of the bacteria, if it happens to be in the environment or is introduced in the environment. So one of the other things that we've been working on is trying to take a closer look at the data. So while we're automating all of these geospatial processes, we're also doing some automated charting of the different variables that we're looking at, trying to identify outliers. And this is going to help us in the next stage of improving our process by allowing us to either weight the outliers differently, or perhaps, depending on how much of an outlier they are, maybe we leave certain data points out. So we're still working on how to handle those outlier values. But one of our goals moving forward is not just having this automated process that's easy for us to improve upon and iterate on, but we also want to make it easier for folks to access the data. So clinicians, other researchers, anybody that's studying the disease, or even members of the public that want to learn more about the disease and spatial variation and risk of exposure to it, to the organism that causes melioidosis. So this, I'm going to show a very quick example of the interactive atlas that we've been working on developing. the folks at Tax have been great in terms of helping us set up some infrastructure that we need to get this off the ground, setting up a geo server specifically that we can use to share out the results that we are generating as spatial services so that we can load those into an interactive map. Our goal with this is to make it really easy to update as new data becomes available. So as more research is conducted, more soil samples, water samples are gathered. We know where the organism exists, or even negative samples where we know it does not exist. We can use that to refine and improve the data that we are in turn generating, the results that we're getting, and we can update the map seamlessly. And it also will give users the ability to adjust the inputs and parameters. So going much beyond just what folks would get out of a single static map that's in an academic publication. And we want it to be simple for folks to use, even if they don't have any geographic information system experience. And we also are thinking this could potentially be adapted to other disease causing organisms or even disease vectors. So I'm going to show you a very quick example of this. This is the interface. Again, it's just a prototype. We're not yet pulling in any real data yet, but the idea is that you'd be able to select the organism you want to look at and assess environmental suitability for. Looking through this map interface, we even want to include the ability to allow folks to model what how conditions might change based on future climate scenarios. And the idea is that folks would be able to toggle on the different variables that they want to look at and basically see an analysis of, and we'd be able to generate that data on the fly because it'd be pre-generated, the results for these combinations of variables so that we can see the results in the interactive map.
You might want to stop sharing and reshare so we can see that last piece.
Yes. Yeah. Give me 1 second. Sorry about that. All right, so yeah, everything I was just describing, I know you didn't actually see because you were still looking at the PowerPoint. My apologies for that. So this is the interface. As I was mentioning, we want eventually the ability for folks to be able to toggle potentially multiple organisms that they'd be able to look at environmental suitability for in the map interface. We also want to include climate, future climate scenarios in our model, and so allowing folks to select different scenarios would be something that would be ideal for us. And then giving folks the ability to toggle on different variables to see how that would adjust and impact predicted environmental suitability for a particular disease-causing organism or disease vector. So I just have one more quick slide before I turn it over. And basically, I just wanted to mention some of our next steps, and some of these I've already addressed. We're still working on improving the script and adding more functionality to it. We want to actually load the real geospatial data coming from a GIS server into the interactive map that I just provided a very quick demonstration of. So bringing that data in as web services to actually allow for all of the functionality that we are hoping to see in that interactive interface. Detecting and managing outlier values. So right now we're starting to implement a process for identifying those outlier values, but what happens when we detect them and how do we manage them? And then adding additional data sets to the model. So there are a few additional data sets that we're looking to add that prior research has shown are in some cases associated with presence of Burkholderia, pseudomallei in the environment. So again, we think that by adding some additional data sets, we might be able to improve the accuracy of our predicted environmental suitability results. And so that's everything I have to share.
Thank you so much. That was great. I think we would probably go to Anna next and then take Q&A all at the end. Yeah, so thank you.
Great. Can you all see my screen? Okay, awesome. Hi, everyone. My name is Anna Dabrowski. I work for the Texas Advanced Computing Center, and Jessica Trologan, also listed here, is my co-PI on our PT 2050 funded Digital Object LifeCycle, also known as DOLCe project. In this presentation, I'll talk about our work on DOLCe this past year invite you to publish your research data products in the Texas Data Repository, and let you know about future work. So first off, the purpose of the DOLCe Initiative is to pursue projects that combine the expertise and the service specializations of the Texas Advanced Computing Center and the UT libraries. And this is in order to develop the necessary infrastructure, mechanisms, and policies for ensuring that data are safeguarded and usable throughout the research lifecycle. So this past year, we focused on a project to improve existing data publication options that are provided by the UT libraries and the Texas Digital Library. Specifically, Planet Texas researchers can distribute their research products broadly with less concern for the size of their digital files that they'd like to make available, and with library stewardship that ensures that the published data will be accessible for at least the next 10 years. Our first project with PT 2050 focused on enabling the FAIR, and like Dave said, that's findable, accessible, interoperable, and reusable publication of large data sets. But we would also like to continue the DOLCe Initiative in other areas, and those include expanding large data publication services to a broader community, improving access to geospatial data, creating shared data collections for computing at TAC, and adding digital preservation workflows for large data. So to start off, our pilot project enables PT 2050 researchers to publish both large and small data sets using the Texas Data Repository publication platform. The traditional repository storage option has so far allowed researchers to publish data sets up to 10 gigabytes in size. And our work has enabled an additional storage option that can be used for data sets that are larger than 10 gigabytes. On the technical side, we've allowed the data repository software to manage multiple data storage locations, one of which is now the Texas Advanced Computing Center, and we've also improved the data ingest process with direct upload for large files. Now we're able to have large files that live on TAC systems, while the smaller files continue living in the traditional storage. Everything is still managed and published online through the Texas Data Repository software, which is hosted by the Texas Digital Library, and all the published data will continue to be stewarded by the UT libraries. If you're curious, further technical details are documented at the link below. And for those who are unfamiliar with it, the Texas Data Repository is a platform that's designed specifically for online data publication and for maintaining long-term access to data files for reusability. It includes a lot of features to enable fair data publication, which aren't guaranteed in other publication venues or by self-publishing through a personal website. Moreover, it's a service that's provided to you with support from the UT libraries, meaning that they can help you go through the data publication process. A few more tidbits about TDR is that it's discipline agnostic, so you can publish your data in any file type from any field of research. The repository will make your data findable, meaning that your published data sets will show up in search engine results, including if people are searching through Google. Your data will be accessible, so the repository provides online access to download data along with descriptive metadata that tell other people what your data are all about. And you, as the researcher, can also designate your terms of use. You can also organize your data sets, so you can build out collections of data sets, each with its own web page. We already have a Planet Texas collection where you can add your data, or you can start your own collection for yourself, your projects, your team, or your lab. Data sets that are published in the repository are citable, so your data sets do receive DOIs that can link directly to them. And the repository also auto-generates citations in several different formats, so if someone's reusing your data, they can also cite your work. Additionally, a neat feature is that your data sets are trackable, so you can update your evolving data sets, and the repository includes version tracking, so you can keep track of the versions of your data, and if someone uses an older version of your data, they can go back and cite that older version, or they can get a new version and see what's most recent. It also includes usage metrics for your data sets, so you can see your download counts. And probably most importantly, it's researcher controlled. So you manage your publication workflows with the flexibility of different user and group permissions to publish your data. And you can choose how you describe and distribute your data online. On the back end, the libraries steward your data, so your published data sets will be accessible online for at least 10 years, with additional back-end data preservation options for the longer term. So as part of our Planet Texas 2050 funding, we have the opportunity to provide PT 2050 researchers with one-on-one support for their large data publication. Everything's now set to go, and we'll work with you and your research team to publish the products of your research in any way that meets your needs, whether this involves meeting the requirements of a journal for access to your data, or a desire to make the data that you've produced available for use by others into the future. We can do this whether the data are currently living at TAC, on your local computer, or elsewhere. We'll help you with describing your data according to standard practices for metadata, and choosing if you need the large data storage or the traditional storage option for your data. In either case, your data will be published and available for download and reuse in the same place. When you're ready to publish your data, visit this link, and please email me and Jessica directly with any questions or concerns. We'll also share the slides after the presentation with this information, so you can go ahead and visit the link from there. I also wanted to tell you a little bit about upcoming work and ideas for additional projects as part of DOLCe. The work we've completed so far to enable larger data publication in the TDR for PT 2050 researchers also affects the broader community of stakeholders. In addition to TAC and the UT libraries, our project directly involves the Texas Digital Library, who host and maintain the Texas Data Repository software. After our pilot with PT 2050, we plan to expand larger data publication services to all researchers at the University of Texas, and we're currently working on policies to do so. Since the TDR is a consortial repository, the software development work and our policy recommendations to the steering committee of the repository directly impact other Texas institutions as well. We'll be enabling them to also implement their own larger data publication options. Additionally, the Dataverse software that the repository uses is an open source project. The code that was written as part of our DOLCe project was contributed back to the general Dataverse project and can be used by institutions across the world who also run this software to manage their own multiple storage options. As Michael mentioned in his presentation, TAC staff are in the process of deploying a geospatial data server in collaboration with PT 2050 researchers. This deployment was based on requests received during our PT 2050 TAC Institute that was held in August. And currently TAC plans to run the open source GeoServer platform. This will allow researchers to share and access geospatial data as Open Geospatial Consortium web services. These web services enable direct access to geospatial data layers using a variety of software and web applications as clients, without users needing to download the data files. This setup can also give researchers fine-grained control over the display characteristics and the manner in which geospatial data are shared. And the preview that Michael gave shows our first use case at TAC. We've been aiming to support the Burkholderia project in their online dashboard that displays map layers. As part of DOLCe, we're proposing to additionally build on this effort and improve the findability and access to geospatial data that's living across different systems. So here you see an early model of the project that we're proposing to pull data from multiple sources, including the Texas Data Repository and the DataX portal into the geospatial data server, and then building out a discovery layer with the data catalog and previewing capabilities. This means that researchers who want to build their own dashboards can still build their own dashboards, but for those who don't and still want to use the geospatial data server, their data can automatically go into a catalog with previewing capabilities. Another effort we've been pursuing is to make large data sets that are published openly online available for research workflows at TAC. Our goal here is to enable researchers to reuse existing data for their projects without needing to deal with transferring the data over to TAC themselves. We're focusing on data sets that are likely to be of general interest to many researchers in the PT 2050 community, which can also reduce the replication of effort by multiple different research groups who are trying to transfer the same data and manage it within their own projects at TAC. So instead, we can make the data available to all PT 2050 researchers in a shared space. The location for the data at TAC will largely depend on the use cases for the data. For example, LiDAR data that we transferred from TNRIST are on the Stampede 2 system because they need to be directly available for computationally intensive command line workflows. We're also transferring soils grids data that Michael mentioned over to TAC. And now we plan to make this available on the Corral system and accessible through the PT 2050 DataX portal as well. Finally, one more project we're interested in pursuing in the future and I'd like to share. The Texas Digital Library is enabling Chronopolis, which is a distributed digital preservation option for data published within the Texas Data Repository within the traditional data storage option. So that's for data sets up to 10 gigabytes in size. We could expand on this and also include the data within our larger storage option at TAC. A project in this area would involve technical work with connecting our storage with the Chronopolis Digital Preservation Network, and would also explore ways to build out additional curation services for large data sets to make sure that they can be preserved for the long term according to best practices and without incurring overly burdensome costs. And finally, I'd like to thank our collaborators and colleagues for their work and support. This includes Courtney Muma at the Texas Digital Library, Jim Myers at the Global Dataverse Community Consortium, as well as Colleen Leon, Chris Jordan, Michael Shensky, Jamie Powell, and Suzanne Pierce. Thank you, everyone. And I think we might have about 5 minutes for questions.
Thank you. We actually have 7 minutes.
Excellent.
Hey, Anna, that was really great. It gives me a lot better understanding, a kind of overview of what you all have been up to. And that seems really exciting, actually. And I think there's a lot of potential here to make scientific workflows more manageable and deal with these large data issues. So that's terrific. So I'm quite intrigued by this GeoServer setup. Would that be, is that something where one can do a range query and pull simple features out across the wire, or how would that be used in terms of if we wanted to get query for and get polygons or other kinds of features out of the database?
I think I might let Michael actually answer that question. I'm more familiar with the deployment of the software and how things get connected at TAC and kind of the policies around that. But I think Michael's more familiar with how this software actually works and how researchers use it.
Hi, Tim. Yeah, I'll say that should be possible. And we are just in the very early stages of experimenting with GeoServer. And unfortunately, it wasn't up and running right now for the very short demo that I did. So I kind of just had to use some filler data sets for that. But yeah, that hopefully is going to be something that the GeoServer will be able to support, because we're also excited about that possibility.
Yeah, nice.
Can you hear me? I have a question. In addition to adding more variables, explanatory variables to your models, are you open to actually create different models with different methods?
Yeah. So definitely, I think we're still in the process of not just refining the model by adding additional data, but also looking at, computationally, are there things that we can do to process that data differently to improve the accuracy of the results that we're getting. And so there's still work to be done on that front, I would say, and there's still some work we need to do into figuring out, specifically what directions we want to take on certain things that we're working on in the model.
Because one easy extension of what you guys have been doing that I think would be more robust would be, for example, to implement weights of evidence type of approaches. That would be somewhat easy to do and maybe get more robust results.
Yeah, absolutely. Weighting is a big thing that we realize that we want to incorporate into the model. Some variables presumably being more important than others. and not weighting them all equally, which is going to give most likely a distorted result that's biased in one direction or another or inaccurate. So that's definitely one of the next steps for us, is looking at how we want to implement weighting in the model to give certain variables greater influence in the results.
I think one of the criticisms of the map that was published about 5 years ago is amongst the people who did it, is that it's probably very over parameterized for what it is. And so there are definitely issues with, if you actually look at the publication, you could say that everything, including the kitchen sink, was thrown into it. And so, and is very highly biased to what people knew about the disease back then, and in only a certain area of the world. So it definitely, we need to kind of, part of it is just pulling these variables apart and understanding which is more significant and should be weighted depending on where you are in the world and what you're looking for. So very, very important point. Thank you.
This is Andrew Payne has a question. Go ahead, Andrew.
Hey, this is probably for Anna. Do you guys have any plans, or where do you see in the data schema you showed earlier where observational patient level health data could be included?
Oh, so that might be a little bit more difficult. I think largely what we're considering here is data that can be shared more or less openly and not sensitive data.
This would be data that had been scrubbed of patient health information, specifically through transforming it to the OMOP common data model.
Okay. Well, I think that, I mean, that would be possible. If there isn't sensitive information, then it definitely is data we can pull into the GeoServer. It kind of just depends on where the data live or where you would like them to be stored, at least from my perspective. But maybe your question's a little bit more research focused.
Yeah, our project, I'm at Dell Medical School. Our project is definitely research focused. And the OMOP common data model has this really elegant philosophy behind it, where the everyone's data is transformed to a common model, but it lives on their site. And data doesn't go to researchers. Researchers bring their analytic to the data where it lives. Because it's been transformed to a common model, an analytic written for any institution's data could work on any other institution's data. But that probably, I mean, that's obviously things get very difficult. You're talking about health data, even if it has been scrubbed at PHI. Okay, cool. Can I see that? Where can we see more about that schema?
For the geospatial data access?
Yes, ma'am.
Right now it's still a proposal, so we don't have that fully fleshed out. So this is kind of a first draft.
Okay, where can we follow its progress?
Oh, I don't really know. Do we have a way of posting that somewhere, Dave? Through PT 2050?
I think we could figure out how to do that. Yeah, let's make a note of that.
Okay, yeah.
Cool. Thank you all.
Yeah, it's slightly past the top of the hour. Thank you all for coming. Thank you for the great presentations. And Jonathan, Allison, thank you for helping put this together. This was great. Reminder from Fernanda at the very beginning of the meeting was, don't forget to vote. So just passing that PSA along one more time. But thank you all. Hopefully we'll see you at the next one of these is October 28th. Is that right, Jonathan?
That's right. Yeah, October 28th, the Wednesday at 12. We'll be doing Fernanda's Texas virtual reality. There will be a project on Texas, on metropolitan Texas, and a project on modeling hurricane resilient healthcare infrastructure. And yeah, well, we have, I mean, if, I know Katie was willing to stick around a couple more minutes. If there is one more question.